Hello everyone, welcome to Science and Tech series with Dr. Vivek Rana. Hope you are doing great. Topic for today's discussion is from biotechnology section. We will be decoding how mRNA vaccines work because UK has become first country in the world to give green signal to mRNA vaccine. This mRNA vaccine is jointly produced by American pharmaceutical giant Pfizer in collaboration with German firm BioNTech. And there's another vaccine which is in frame by Moderna in America, and both are showing promising results with efficacy as high as 90 to 95%. And the common thing between both these is are based on mRNA. Till now, there has not been a single vaccine which has been clinically given approval to be used on humans. So this makes it a landmark of first that mRNA vaccines will now be a reality. So from examination point of view, we'll be decoding how these mRNA vaccines are different from the conventional vaccines. So let's just decode how mRNA vaccines work. But before we come to the main picture, we need to brush up few biology themes. Only then you will be able to understand that what exactly is happening in these so-called next generation vaccines. So in our body, we know the nucleic acids are of two types. There is DNA and RNA. The DNA is present in nucleus, is a double helical structure. So this is what I have just depicted. Here is the DNA which codes information in RNA. RNA then uses this information to code for amino acid sequence, which ultimately forms a protein which can be used as an enzyme or an antibody. Technically, this process is described as transcription and translation. When DNA transcripts information to RNA, it is called transcription. When RNA synthesizes proteins through ribosomes, then it is called the translation process. So you have to understand they work in collaboration, and this is used in multiple physiological reactions. And then coming to the normal vaccines, in vaccines mainly work against an antigen. Just like some bacteria or virus enters our body, our immune system recognizes them and produces specific antibodies. These foreign particles are called antigens, whereas our useful proteins against those foreign particles are called antibodies. So in vaccines, we introduce the weakened strain of viruses. Either the part which is causing the infection is weakened, or at times the dead bacteria or virus is introduced first to prepare our immune system to fight against it. So this is how the conventional vaccines work. Either they are live attenuated vaccines or the dead ones. Now conventionally, all the vaccines which are currently working are in a way introducing the weakened strain of viruses or bacteria, and then our immune system responds to them. So in case of mRNA vaccine, there is no such concept of introducing a foreign antigen, because there is a risk when you are introducing such weakened viruses, they might revert back to their original pathogenicity and they might cause infection. Even though the cases are almost negligible and that risk we take overall, but in case of mRNA vaccine, there will never be a case because no such virus is being introduced. So coming to the main theme, that how these mRNA vaccines are working, and make sure you are also clear with the word M. M stands for messenger RNA. The RNA are of three types. There is a tRNA, there is mRNA, and there is a rRNA. So currently, out of those three, the messenger RNA is gaining limelight, and because it is in a way acting like a messenger of DNA, it carries the message to different parts of our body to produce specific proteins. So in mRNA vaccines, what we are doing is that we inject the mRNA in intramuscularly in our body. Just as you can see in diagram, the vaccine prepared will be injected through an intramuscular route, and once the mRNA enters, it is genetically programmed to produce specific type of proteins, and those specific proteins are none other than the spike proteins. The current virus which is making the pandemic involved in the pandemic is the SARS coronavirus 2 strain, which is characterized by the spike proteins, and that's what is the reason it is given the name corona, because in a way it is resembling the royal crown, or you could say sun corona. Okay, so we can say that the main reason the spike proteins are to be produced, because this is the way through which it is attacking our you could say the lung receptors. The spike proteins get attached, and then the virus replicates and produces multiple copies, which harms our human you can say the lung cells, and that results in the pathogenicity. So here what we are doing is this mRNA is being programmed to produce this specific spike protein, and once the spike protein is produced, our immune system gets activated. It produces the specific antibodies. We all know in our body the white blood cells are the soldiers of body's defense system, and the specific type of white blood cell beta cells produce the antibodies. So these Y-shaped protein are our main defense mechanism. They will come into the picture and recognize it. So basically then the booster dose is given after 2 to 4 weeks, and then you can say our immune system will be ready to fight the battle. So in case the coronavirus, even if it enters, our immune system would be ready and it will just counter it effectively. So this is how in this case there is no such introduction of the virus. Here we are programming it to produce the main ingredient and we're trying to produce that spike protein which will prepare the body to fight it once the actual virus enters. So comparatively these are safer vaccines. On the downside we can say this mRNA is a fragile molecule, it easily disintegrates, so that's where we have to just bind it with the fat molecule and store it under minus 35 or minus 70 degrees. Moderna and Pfizer vaccines vary in their cold storages, and that's what logistic challenge is going to be. But nevertheless, this is an effective milestone in, you could say, biotechnology. Now we can say there is another set of vaccine which can be prepared, and this treatment will just not be limited to fighting the coronavirus. This mechanism can be used for other viral infection or some other treatments where we are still to reach the efficacy which we desire. So it is quite a major milestone movement in field of, you could say, physiology and chemistry, and the persons who have really worked hard on this mRNA, they have been working since 1980. They are finally getting their recognition, and already the calls have been made that you might see in couple of years or even in next decade the Nobel Prize being awarded for this mRNA vaccine discovery. So it's a major breakthrough, and hopefully it will be better for the world as soon as these vaccines get, because UK has decided to just roll out within the next week, and we will see more countries follow the suit. So these trials, even though have been carried against the time and have been streamlined to get the approval, but nevertheless an important milestone, and it was required. So if the world gets together, we can definitely fight this pandemic and emerge victorious. So I hope you get a fair idea of what mRNA vaccines are. Make sure if you have any specific doubts, you can ask them in comment section. I will try to answer them, and you can revise the different types of RNAs, how nucleic acids are formed, what are their building blocks, the basic concept of antigen and antibodies, and how are the conventional vaccines like live attenuated vaccines different from these mRNA vaccines. So this is what we have in today's Science and Tech Update. Thank you very much. Have a nice day.